It's my pleasure to introduce the next winner of the Early Career Award. This one is typically given to the young researchers in the bioinformatics or computational biology who completed their PhD in the last 6 years. And this year winner is Michael Skinnider. And he was selected because of his great achievements in the very different fields of bioinformatics, and also by his support or maybe contributions to the bioinformatics community. A few words about his career. So he actually started with the single-cell RNA-seq data. He did nice benchmarking of the methods for differential expression analysis there. He also came up with the machine learning approach, Augur, to identify specific cells which are critical for the healing process after walking issues. He also then moved to mass spec data, to proteomics, and later to the metabolomics. And there he developed a new, let's call it metabolic language model, DeepMet, to dive into the to the dark matter of the metabolics data and identify the new or until now not known metabolites in the spectrum. For the community, he also developed several R packages, and from the other achievements, he also helped to defend and maybe rescue the MD PhD program in Canada. And as a last kind of piece of information, he was offered a faculty position at the Princeton University directly after a PhD. So please welcome to the stage, Michael Skinnider.
Thank you very much for that very kind introduction. I want to start just by saying, how grateful I am to be speaking to you today. This is really an enormous honor, and it's made more meaningful for me by the fact that many of the previous recipients of this award are people who I've really looked up to and whose work has been very meaningful and inspiring to me. So I thought I would tell you a little bit about our work on developing computational methods to help discover previously unknown mammalian metabolites. Before I jump right into that, though, I thought I'd just talk a little bit about my own background and how we came to be interested in working on this particular problem. I am, of course, a computational biologist by training. That training really encompassed many different kinds of biological and biochemical data, from genomics to transcriptomics to proteomics, in addition to metabolomics. So, for instance, I got my start in research as an undergraduate student at Hamilton, or a university in Hamilton, Canada, where I worked with Nathan McGarvey, and my work there focused on developing computational tools to discover bacterial secondary metabolites, or natural products. These methods used as input genomic and metabolomic data sets. And so this is where I really first developed a deep interest in metabolism, and not just metabolism, but in technologies for measuring metabolism. Notwithstanding that interest, when I moved to the west coast of Canada to do my MD PhD, I also moved into a different field. So my work with Leonard Foster focused on mapping protein interaction networks using a technique called cofractionation mass spectrometry. And this work culminated in the first tissue-specific protein interaction networks in mammals. During that time, I also spent quite a bit of time at EPFL, where I worked in Gregoire Courtine's lab, developing computational tools to identify subpopulations of neurons that produce a particular behavior, this time using single-cell and spatial transcriptomics data as input. And ultimately, these methods pointed us towards a subtype of spinal cord neurons, which can be activated through electrical stimulation of the spinal cord in order to restore walking in human patients who were previously paralyzed. At Princeton, as I said, my lab has really come full circle to my undergraduate interest in metabolism. But now, instead of focusing on bacterial secondary metabolism, we're really much more interested in the mammalian metabolome, and particularly in discovering metabolites associated with the development, progression, or treatment of cancer. So I tell you all this in part to explain what it is about metabolism that I just could not stay away from. You know, I told you about very different themes and ideas in genomics, transcriptomics, and proteomics, and these are, of course, three very different fields. But I would argue that they all have one important thing in common, which is that in these fields, it's become routine to measure essentially the complete complement of DNA, RNA, or proteins in any given biological sample. And the situation in metabolomics is really very different. It is still today essentially impossible to comprehensively identify the small molecules in that same sample. So this seems like a very profound gap in our ability to study and understand living systems. It's a gap that I think is particularly exciting from a computational perspective, because I would argue that this gap is not for lack of an appropriate experimental technique. In fact, with mass spectrometry, we already have an analytical technique that can detect or acquire data from thousands of metabolites in any given biological sample. And not only that, but mass spec collects very rich and multifaceted data for every one of these small molecule metabolites. So this seems much more like a computational problem. How can we take this rich mass spectrometry data and deduce from it the structure of the metabolite that was observed by the mass spectrometer? And that, unfortunately, turns out to be sufficiently difficult that today, in a routine metabolomic study, really only a very small fraction of the signals that are required by the mass spectrometer will actually be identified, that is to say, linked to the chemical structure of the corresponding metabolite. And so the rest of that data is commonly referred to as the chemical dark matter of the metabolome. So this dark matter is really what we're focused on illuminating. We're broadly interested in using computational tools, many of them leveraging advances in AI, to discover this hidden chemistry that's sort of hiding in our routinely collected metabolomics data, and really do that as a means to finally put together a complete map of the mammalian metabolome. So that is quite a grand challenge, and you might ask, how can we possibly make progress on this? I'll tell you a little bit about the logical starting point for an approach that we've had a lot of success with over the past 2 years or so. And that thought is sort of as follows. Over the last century, scientists have amassed an incredibly rich understanding of the known mammalian metabolome. So can we now leverage that understanding in a systematic way to predict the composition of the as of yet unknown metabolome? And the computational approach that we've been using to explore this idea is based on chemical language models. So the core concept here is that we represent the chemical structures of known metabolites as short strings of text. We particularly like a format called SMILES that I'm showing you here. And of course, we do this because representing metabolites as text allows us to repurpose the same kinds of neural networks that have been so successful at learning the syntax and semantics of natural language. And instead of applying these language models to the words in a sentence, we can instead effectively apply them to model the atoms and bonds in a chemical structure. And so our hope was that this approach might allow us to, as I said, learn from the structures of known metabolites to generate structures for as of yet undiscovered metabolites, which we could then target for discovery experimentally. I should mention that this was an idea that we already had a certain amount of proof of concept for in a related field, specifically that of forensic chemistry, where we had previously used language models to help discover emerging illicit drugs of abuse, also known as designer drugs. So a few years prior to this work on the mammalian metabolome, we had trained a chemical language model on the structures of all known designer drugs, a model that we called DARK-NPS. And after we trained this model, we did something a little bit unusual in the field of machine learning, which is that we waited. We actually waited for 6 months, and over that period, of course, new drugs of abuse were emerging on the market around the world, and they were being discovered by forensic laboratories. And so at the end of that 6-month period, we asked how many of these emerging drugs of abuse did our language models successfully anticipate? And remarkably, we found that dark NPS successfully generated more than 90% of these emerging illicit drugs in this prospective test, including the structures that I'm showing you here. We then also showed that we could integrate the language model's predictions with mass spectrometry data, and that this allowed us to discover previously unknown illicit drugs in clinical samples, as well as in law enforcement seizures, with really a remarkable degree of accuracy. And in fact, we worked with the Danish National Forensic Lab to discover or help discover a new dissociative drug, this derivative of the well-known street drug PCP that I'm showing you here. So all of this, past work really led to a lot of optimism that we could use essentially the same approach, and rather than learning the medicinal chemistry logic of illicit drug synthesis, we could instead learn the biosynthetic logic of metabolism. So we implemented that idea in a chemical language model that we named DeepMet. DeepMet is a model that's been trained on the structures of all known human metabolites, and which, as I'll show you, can guide the discovery of novel mammalian metabolites. And much of the work that I'm going to show here was led by Tony, a PhD student in my lab. So I mentioned that we had trained DeepMet so that we could generate new metabolite-like chemical structures, which we could then target for discovery. And we found that DeepMet did an excellent job of generating metabolite-like structures. It's so good, in fact, that a second machine learning model could not tell apart the generated structures from those of known metabolites. And just as we had seen with dark NPS, we found that DeepMet successfully generated the vast majority of newly discovered metabolites that were added to metabolic databases after we had trained our model. These predictions, in fact, were good enough that we could buy or synthesize chemical standards for compounds that DeepMet predicted ought to exist. And then we could use the data that we'd acquired from these standards to actually discover these metabolites in human samples. So here I'm showing you two examples of metabolites that DeepMet predicted likely existed as human metabolites, which we were able to experimentally confirm in human urine. We also, again, developed approaches to integrate these predictions more directly with metabolomics data so that we could assign structures to many of the unidentified peaks or signals within the mouse metabolome. So for instance, here I'm showing you a mouse metabolite that's very specific to the kidney and pancreas. We synthesized the predicted structure shown at the far left, and we found that the experimental data from this chemical standard matched almost perfectly to the corresponding signal in the mouse kidney. This is an approach that we've now been able to scale up to some extent, to the point that we've now used DeepMet to discover more than 40 previously unrecognized mammalian metabolites. And this is really exciting to me because it suggests that we could really use this technology to accelerate the mapping of the mammalian metabolome. So, why does this approach work so well? I think a good explanation is that chemical language models effectively allow us to narrow the search space when we're hunting for a particular category of small molecule. Chemical space as a whole is sort of incomprehensibly vast, but metabolite-like chemical space or designer drug-like chemical space are small subsets. And so by narrowing our search to consider only metabolite-like structures, we've essentially made this problem a lot easier. That being said, I think that there are still really important opportunities to do a better job of linking these generated structures to the mass spectrometry data itself. And so this is really a major focus of the lab going forward. We have a few different ideas about how to do this that are now underway. For instance, collecting more data ourselves, learning from unlabeled data, for instance, with self-supervised learning, learning from new types of data that are not really typically considered in structure annotation in metabolomics, and then, of course, simply developing better machine learning models. So I'll stop there, maybe just by reiterating this idea that our inability to comprehensively measure small molecules in biological systems is, I think, an opportunity to address a fundamental challenge using computation, and this is really the challenge that motivates my lab. So I'll stop just by thanking the people in my lab who worked on this, of course, our funding sources, and once again, I really want to express my gratitude to the Swiss Institute of Bioinformatics. So thank you.
Thank you, Michael. Congratulations. Any question? The audience? Yes, please.
Yeah, thank you very much for the interesting presentation. I like the test that you did, just waiting for design of drugs to be created and see whether or not you could have detected them. And 90% is quite impressive. But then my question automatically is, actually, how many of them did you generate? And linked to that, since those are LLMs, I guess you are able to rank which are the most likely also in terms of metabolites.
Yes, those are all, that's a great series of questions. I sort of, simplified for the purpose of just time, but in fact, we do, assign a score to each of the generated structures that sort of correlates well with its likelihood of occurring in the future. So the 90% number, it reflects the proportion of generated structures that were detected, but the denominator here is huge. It's on the order of millions. That being said, in both that work and then in the DeepMet work, we found that we could actually use that score to sort of prioritize without any analytical data at all which of the generated structures are most likely to emerge or be discovered in the future.
And so I imagine that's how you're trying to detect the new metabolites, just looking at the most likely ones.
Exactly, and then using that same idea to sort of re-rank our interpretation of the mass spectrometry data, particularly the MSMS data itself. Yeah.
Sorry, so our schedule is quite packed. We don't have time for another question. I think that you could discuss during the lunch.
Thank you.
Thank you again, Michael.